Okay, hello everyone. We are going to start off quickly because I want to make sure we get through the rest of our material on the GI system today because we have very few sessions left. And I want to make sure we spend at least one day on the male reproductive system and one day on the female. So whatever we don't get to with the GI system today will not be on your exam. I will try to send out an email about where we got to for anyone who's not here today. Okay. Sorry, I was answering a question in the chat and letting a couple more people join us. Okay. So when we left off, we were talking about GI absorption. We had gone through the basics of carbohydrate digestion and a little bit about protein digestion. So just as a reminder, different computer. Okay. Okay. So when we were talking about carbohydrates, we saw that they start to be digested in the mouth with saliva, specifically using salivary amylase. And then these carbohydrates that have now been slightly broken down continue through your esophagus, into your stomach, into your duodenum, where they mix with the secretions coming from the pancreas, so the pancreatic juice. One of the enzymes coming from the pancreas is pancreatic amylase, which continues the digestion of the carbohydrates, breaking them down into smaller little carbohydrates, disaccharides and limit dextrins, which then have to be broken down into monosaccharides to actually get absorbed through the epithelial border. That usually involves enzymes we're not worried about here that are embedded in the walls of the epithelium itself. So proteins, in contrast, don't really start to get broken down in the mouth. The proteins get broken down more when they get to the stomach. So we have a couple different classes of peptidases, things that break down proteins. So we had our endopeptidases, our exopeptidases, and our inactive zymogens. They come into play in the stomach, starting with pepsinogen, which is an inactive form. So that would be our zymogen for the enzyme pepsin, which is what we get after it's activated using the acid in our stomach. Okay. Our chyme then moves into the lumen of the small intestine, so into the duodenum, where it's joined by fluids, right? Our pancreatic juice coming from the pancreatic duct. That includes a bunch more enzymes for breaking down proteins. So we have pancreatic proteases. We got 3 of them we're interested in. And we have some enzymes in the walls of the lumen of the small intestine that help activate these from their zymogen form into their kind of active enzyme form, where they can then continue the breakdown of proteins in the small intestine. Okay. So absorption of the amino acids has to occur across the wall of the lumen, right? The wall of the small intestine. So the proteins are going to get into the body, usually either linked to sodium absorption, and usually linked to sodium absorption. We have basically different types of transporters for every one of our 20 different amino acids, as well as for some of our dipeptides and tripeptides, which are respectively 2 amino acids linked together or 3 amino acids linked together. So all of these amino acids and the dipeptides and tripeptides are crossing into the apical membrane by active transport, usually secondary active transport. They then, if they're still big, if they're still a dipeptide or peptide get broken down a little more inside the cell, but all of our amino acids then in their actual individual monomer forms cross the basolateral membrane by facilitated diffusion, so down their gradient through a transporter in their monomer form into the plasma. So that is about where we left off. So what we're going to really start out with today is our lipids. So lipids have a kind of special problem for absorption in that lipids are nonpolar, right? So we're eating these mainly in the form of fats, right? Maybe some oils. So the lipid digestion is going to look a little bit different because these lipids aren't going to really mix well with water, mix well with the contents of the lumen, so the contents of these fluids moving through the intestine. A typical diet includes about 50 grams per day of lipids. 90% of that is triglycerides, so that's really what we're going to talk about as we talk about lipid digestion today. Yeah. So because our lipids are nonpolar, they're going to be forming these things called fat droplets. We're going to have to break down these fat droplets in order to get the lipids into kind of a small enough form and have enough surface area that our enzymes can actually get to the lipids and start to break down those triglycerides. The enzymes we're trying to give access to our lipids, to our triglycerides, are mainly coming from the pancreas. We do have some enzymes in saliva, but they're very limited. So most of lipid digestion is happening using our pancreatic enzymes, specifically our lipases. So lipases break down lipids. But they can only act on molecules at the edge of a fat droplet. They can't get into the center of the fat droplet. They can't muscle in there. So we're going to need to break down our fat droplets, which are clustered together because lipids don't interact well with water. So they're all basically get attracted to each other because they're repelled by the water. We're going to need to break down these big fat droplets in order to give the lipases from the pancreas access to the triglycerides so that they can start to break them down. Okay. And this is where bile salts coming from our liver come into play. So here we have a picture of a fat droplet, right? So we have our fat droplet here. It's nonpolar. What we are going to be trying to do is to break it up, right? We want to break it up into chunks so that there's a lot of surface area, a lot of outside layer for our lipases to attack and be able to break down. So the special property of the bile salts is that they are amphipathic. So you've heard this word before, but it might have been a while. So what we previously described as an amphipathic molecule was our phospholipids in our cell walls. So amphipathic means that they have both a hydrophobic, nonpolar, and a hydrophilic polar side, which means that they're able to basically touch water and touch nonpolar things, in this case, our triglycerides, our fat droplet. So what bile salts are able to do is emulsify the fat. And so what that really means is that they're able to break it apart and allow it to mix in with the water a little better. And an emulsifier is something that kind of lets nonpolar and polar things live together a little better. So here we can see a hydrophilic group, so these polar groups, and our hydrophobic groups here. So, but what we really want to do is zoom in on what the products we're going to get as these bile salts come in and break down our fat globules. So we'll take a look here. So here we have our original fat globule at the top. So our original fat globule right, is totally nonpolar. So it doesn't have any of those polar groups on the outside. It's repelled by water, right? So all our fat has kind of glommed together. Then comes into contact with these bile salts, which are amphipathic. So we can see their polar groups and their nonpolar groups. And this allows the fat globule to break apart. So in this case, we've kind of broken it into like three. There we go, broken it down into three smaller little fat droplets, which means that as they're smaller, we have more surface area for digestion with our pancreatic lipases. So now we're thinking about these smaller fat droplets. Okay. So the fat droplets are mainly made up of our triglycerides. So we can see our triglycerides. We might have some phospholipids in there. We might have some other types of lipids in our fat droplets as well. But we're focused on the triglycerides today because we're mainly eating triglycerides in the form of fats and oils. So some of these fatty acids are going to be absorbed. Basically, when we absorb fats, we're going to absorb them in the form of individual fatty acids or these structures called monoglycerides, which is basically if we take our typical form of a triglyceride is a glycerol and 3 fatty acid chains, right? That's why it's a triglyceride. What we're going to do as we break down our fatty acids, as we break down our triglycerides, we're going to like break off fatty acid chains from the end. So drawing those in green now. And that's going to leave us with this kind of T structure from the center. And that's our monoglyceride. So these are the forms that are kind of small enough to get into our cells. Okay. So some of those are going to be absorbed immediately, like as soon as we have these pancreatic lipases break apart the triglyceride. Others are going to move from these fat droplets, and they're going to form these structures called micelles, which we'll take a look at in a second. The micelles are basically going to be clusters of these broken down individual fatty acids and monoglycerides. They're going to be floating around in the lumen, and we're periodically going to have little pieces breaking off of these micelles. And by little pieces, I mean some of the fatty acids and monoglycerides will break off. And we're basically going to form an equilibrium state between the fatty acids and monoglycerides remaining in the lumen and the fatty acids and monoglycerides in the absorptive cells. So as they move through and as they move past new cells that don't have these fatty acids and monoglycerides in them, more and more pieces of the micelle are going to break off. So let's take a look at that. Sorry, okay. So here we see a zoomed in picture of some of these structures. So here's our fat droplet. We can see it's got some bile salts attached to it. So it's been broken down from that big fat globule already. Okay. So here we see some lipases come in, and we can see that we're breaking down a triglyceride kind of into its constituent parts here. So that's what these are, right? We have our monoglyceride, kind of T-shaped. We have our individual fatty acids here. And here's what a micelle is, all right? So a micelle is the structure where we have kind of the fatty acid kind of legs of things pointing in towards the center. And if they have any polar parts like these phospholipids, those will form towards the outside. But this is much, much smaller than our fat droplet, right? And it's just consisting of things that are small enough to move into a cell. So that's the difference between the micelle and the fat droplet. The fat droplet has our big triglycerides, right? So the bigger kind of lipid forms. So from these micelles and from these broken down triglycerides, right, are monoglycerides and fatty acids, and then the micelles that are formed of those just kind of circling up. These are small enough to move directly across the phospholipid bilayer into our absorptive cells, so into the cells that are absorbing our nutrients, basically. So they don't need a transporter because they're nonpolar, just like the cell membrane is nonpolar. So they can just move directly across just by simple diffusion. So remember, simple diffusion is moving directly across the membrane without need for a protein, without need for a transporter. Once they are inside the epithelial cell, so inside that absorptive cell, there's going to be a little more processing. They're going to basically reform some larger lipid structures before they move into the plasma. So the triglycerides are going to get reformed inside the cell once we move these small parts in. And then these lipids are going to move into the Golgi, right? So here we have our first organelle that they're entering, and then they're moving into the Golgi apparatus, our second organelle that they're entering. They're going to be packaged into this thing called a chylomicron. A chylomicron is basically kind of a combination of lipid and protein. And then these chylomicrons are what actually enters your body. Uniquely, so unlike the carbohydrates and the proteins, these chylomicrons, so these lipids, are going to move into the lymphatic system instead of directly into the plasma. So they're going to bypass the blood, they're going to move into the lymph, they're going to move through the lymphatic system before they're eventually dropped into the venous circulation to move into the heart, and then they will mix with the blood. So we can look, I think we have a picture of that coming up. Let's take a look. Yeah, we do. Okay. So up at the top here, we see our monoglycerides and fatty acids, which might be coming directly from a lipase breaking them off of a fat droplet, or might be released by the micelles as they pass through fresh new cells that create a gradient for the small lipids to travel down. So they're traveling into the cell directly across the membrane. They're then moving into the endoplasmic reticulum. It's actually specifically the smooth endoplasmic reticulum, which we think about kind of way less than the rough endoplasmic reticulum. So we now finally see its role after, I don't know how many years of biology you've had, but we haven't really talked about the smooth endoplasmic reticulum in many of the classes I've taught until now. So they're in the smooth endoplasmic reticulum, reforming the triglyceride. This triglyceride then gets passed to the Golgi apparatus, where we form the chylomicron. Then the chylomicron is what's exiting the cell by exocytosis, skipping over our capillaries, so skipping over the cardiovascular system and moving into the lymphatic system, into this lacteal, right? So we talked about the structure kind of the small intestine, right? We have this brush border of the apical membrane, increasing surface area, but we also had these kind of like projections overall. So this would all have a brush border. And we had our little arteriole flowing into it. We had some capillaries. We had our little venule exiting it. And we had that lacteal in the center, right? So that is what that was there for. The lacteal is specific for the absorption of chylomicrons, which is how we're absorbing our lipids. So this is just kind of showing you that the bile salts do get reabsorbed by the capillaries, so they kind of get recycled by the body. So they return to the liver, where they're then secreted again into the bile eventually. that happens to be called enterohepatic circulation, but I don't expect you to remember that word. Okay. So that leads us to the absorption of vitamins. So we've gone through our 3 big types of macromolecules that we have in food. We're not too worried about digesting a lot of DNA, right? We're mainly absorbing carbs, lipids, proteins. But vitamins are super important to the function of your body, even though they don't constitute that big of a percentage of your food. But we are interested in how they get absorbed, because if you don't have vitamins, you're going to be in trouble. Okay. So big difference that we're interested in when we talk about vitamins is the difference between fat-soluble vitamins and water-soluble vitamins. So this will come back in your future careers, I expect, because fat-soluble vitamins and water-soluble vitamins have different considerations when you're thinking about how long they're going to stay in the body. And they also, here, as we're talking about the GI system, have differences in how they're getting absorbed by the body. So water-soluble vitamins are going to require special transport proteins, and they're also going to get excreted more quickly, right? Because they're water-soluble, so they mix in well with urine. They're generally in your cardiovascular system, right? They're circulating around a lot more. Whereas fat-soluble vitamins are going to get absorbed with the lipids, and they tend to stick around in your body longer. Okay. So our fat-soluble vitamins are vitamins A, D, E, and K. They're getting absorbed with the lipids, so you would expect to see vitamins mixed in with those lipid droplets. You'd see them mixed into the micelles, and then you'd see them packaged up with the chylomicrons. So you don't have to remember a specific separate pathway for them. Just assume they're kind of mixed in there, but not drawn in on the diagrams. The water-soluble vitamins each have special transport proteins that allow them to be absorbed. We talked about one of them already. So for our example, we're going to pick vitamin B12. So vitamin B12 is a vitamin that needs to be bound to intrinsic factor. So remember, intrinsic factor, where we mentioned it before, was when we were talking about the secretions of the stomach, right? So intrinsic factor is one of the things that the stomach is secreting. The reason it's important is because in order to have these special transport proteins allow vitamin B12 to be absorbed, it has to be bound to this intrinsic factor, which is basically a protein. So we're also interested in absorption of minerals. When we say minerals, like usually we mean a lot of the ions that we've talked about throughout this course. So we mean sodium, we mean chloride, we mean potassium. And these are absorbed basically in pretty similar ways we've seen in other systems. So sodium is absorbed by something called solvent drag. And all solvent drag means is that as water is being reabsorbed, it kind of pulls the sodium with it. So they're moving together, they're being reabsorbed together. So that's usually happening in the duodenum and the early parts of the jejunum. So kind of early on in our small intestine. Then we're going to have some active absorption. So some actual like active transport. So some pumps basically in the jejunum and the ileum and the colon. So the later parts of the small intestine and then into the colon. Chloride just tags along with sodium, right? So that negative charge follows the positive charge of sodium. Pretty simple. That's what we've seen it do elsewhere. And we never really focus on it because it's usually just kind of following sodium. Potassium is mainly being passively absorbed. So kind of like this solvent drag. we have some passive absorption of potassium moving down its gradient as it moves through the small intestine and then the colon. What's interesting about potassium is that when concentration of potassium in the lumen, so in the food moving through your colon, is really low, potassium does actually get secreted into the lumen. So it moves back out of your body and into feces, basically. So this can cause alterations to the balance of potassium in your body, right? So like we talked, shouldn't say we talked about it, we mentioned that hypo and hyperkalemia exist, right? So if you're secreting potassium into the lumen, this could be one of the reasons for hypokalemia. Bicarbonate is our next topic. So we've talked about bicarbonate as it relates to the GI system when we talked about the fact that bicarbonate kind of needs to buffer out, so neutralize the stomach acid as it moves into the duodenum. So we have bicarbonate being released there. It's in like our pancreatic juice, right? One of the components of the pancreatic juice is bicarbonate. So that's where it's moving into our system mostly, is neutralizing our stomach acids. And then we're going to absorb the bicarbonate as well. So after we secrete it into the duodenum, we're going to start to absorb it into the jejunum, right? We want to kind of have that bicarbonate back in our body so that we can reuse it. So that's happening passively in the jejunum. When you get to the ileum and the colon, sometimes we're going to see secretion there. And the secretion will be kind of coupled with an exchange of chloride ions. But mainly the thing we want to remember about bicarbonate for the GI system is that fact that it's neutralizing the stomach acid rather than its absorption. Okay. Calcium gets actively absorbed. So that's happening again early on in the small intestine, right? We said most of the absorption of nutrients in food happens in the first kind of 20% of the small intestine. This is why we keep seeing the duodenum and the jejunum popping up. So calcium absorption is active. It's binding to a protein on that brush border, so on that apical membrane with all its projections. The protein is very conveniently called calcium binding protein. And then it gets transported into the epithelial cells. To get out of the epithelial cells, there's a calcium pump. Iron, we talked about when we talked about the life cycle of red blood cells. So as you can see, a lot of these are kind of drawing way back on our understanding of material previously in this course. So iron, we remember, is one of the things that we get as we start to digest red blood cells, right? As we start to break them apart, right? We're turning them into like bilirubin. We're releasing some iron. We want to reabsorb that iron so that we can reuse it to make more hemoglobin, basically. So in order to reabsorb iron, we need to use this molecule transferrin, right? So it's transferring the iron. Anytime you see this F-E-R-R, that's a kind of Latin root that means iron. So it's transferring iron. This is secreted, transferrin is secreted by the enterocytes. So the enterocytes are one of the specialized epithelial cells of the small intestine. So they're secreting this molecule transferrin into the small intestine. Once we have transferrin in the small intestine, it binds to the iron, forms a transferrin iron complex. So when we saw it before, it was kind of listed as T iron. Sorry, I'm drawing on my second monitor. Is why my eyes keep going that way. Okay, so we bind to the T iron. Once we're bound to T iron, we can take the iron this sort of T-iron complex into the cells. We use a specific receptor protein for this. So a receptor embedded in the walls of the small intestine, so protein. So our T-iron would bind to this receptor, which would then trigger endocytosis. So the kind of folding in of the membrane, forming a bubble, forming a vesicle, moving that T-iron in. Once we have absorbed this iron, which is sometimes something we've eaten, but sometimes a product of digestion of hemoglobin, we're going to store the iron. So when we saw it before, we were storing the iron in the liver. When we're talking about iron that is being absorbed from food, it's going to get stored sometimes in the enterocytes. When we're storing the iron, we call it ferritin. Okay. Ferritin, there's that root again. So we saw this before listed as F iron, I think. So that's iron kind of turned into ferritin, kind of balance of ferritin. Some of that iron will then move into the blood to make more hemoglobin or for other uses in the body. It'll be released as needed, basically. When we move the iron out of the enterocytes and into the blood, we're going to need to bind it to transferrin again. So transferrin is always kind of a car for iron. So finally, we are going to be absorbing water as well from the food we eat. So water absorption is passive. We do also secrete water in the GI tract. So we have kind of this balance coming back and forth. Water absorption is following the absorption of solutes by osmosis. So water would be following those other minerals, those ions we've talked about previously. So like when we said there was solute drag with water with the sodium, so that's one of the places we would see water reabsorption. And in general, we'd kind of see some more compaction as we move further through the colon. So we might see some water reabsorption there. But when you think about water, I really mainly want you to be thinking about kidneys when we think about water balance. It's true that the GI system is involved in the absorption of water, but we're not going to go into detail about water balance with the GI system the way we did for the urinary system. Whew. Okay, so that moves us to our final GI topic, which is control, secretion, and motility. So I'll give you a second to pull this up. So as we talk about this, we're going to be talking about how the nervous system relates to releasing these various enzymes in order to break down food. And we'll also be talking about motility, which is like the physical movement of food through the gastrointestinal tract. So we are going to have a number of different kind of hormones involved in this, and sometimes we're going to talk about them in terms of reflex pathways, some of which are short and some of which are long. So we'll kind of dive right into the different phases as we talk about this. Okay. So we talk about phases of gastrointestinal control. What we're really talking about there is like where our signal to do something, basically where our signal to release hormones, to release enzymes are coming from. Okay. So we have a cephalic phase. So like a cephalic phase is when you think like, oh, I'm hungry, right? So cephalic phase is going to come from stimuli originating in the head. So like when you think about food, when you taste food, when you smell food, that all has to do with sensory receptors elsewhere in the body, right? Like not in your GI system. So that would be the cephalic phase of gastrointestinal control if we're talking about how a hormone or something is released in response to these kind of visual or olfactory or even gustatory, like taste senses. Okay. So when we look at cephalic phase control, we're going to expect to see input from the central nervous system. We're going to refer to these reflexes as long reflexes. Actually, we're not really going to go into detail on the long or short reflexes. All right, we've got enough material coming up here, so let's ignore long and short. Okay, let's cross those out. Let's just understand kind of the difference between these phases. So cephalic phase, coming from your sensory organs in your head. Gastric phase is when we have some stimulus in the stomach itself that is causing us to release hormones. The intestinal phase is when the stimulus is in the intestines. So gastric phase and intestinal phase, the stimuli are going to be something like your stomach is really full, or your stomach is really empty, or your intestines are really full, or your intestines are really empty. So something kind of local like that is what we'd be referring to for stimuli for gastric or intestinal phase. Okay. So the reason we're interested in these three kind of phases, the reason we're interested in our hormones overall is largely how they're going to aid in digestion. And so when we think about digestion, we should be thinking about the enzymes that we're releasing. The enzymes are going to be in saliva, they're going to be in the stomach acid, or stomach acid is part of digestion itself. Pancreatic juice, bile. We've seen how these are important products that help us actually break down food, digest it, and ultimately absorb it into the body. So when we're thinking about the regulation of secretion, that's where we're headed, right? So saliva coming from our salivary glands in the mouth, acid, so hydrochloric acid, and pepsinogen, that zymogen that turns into pepsin to break down proteins in the stomach. Pancreatic juice coming from the pancreas, bile coming from the liver, and then ultimately we're also going to talk about the actual movement through the digestive tract. We're going to start with saliva. So saliva, we're going to think of these stimuli as kind of being more cephalic in nature. Saliva secretion is going to happen largely in response to the taste and texture of food, so in response to actually eating. So when you actually eat, this stimulates mechanoreceptors and chemoreceptors in the mouth. So receptors actually feeling the food would be the mechanoreceptors. Chemoreceptors would be sensing the different chemicals, the different the different types of macromolecules in your food. So we're going to taste and feel the texture of the food in our mouth, and that is going to relay a signal to the brain, to the medulla specifically, which has a salivary center. So there's a little integration center in the medulla, which is going to regulate parts of the autonomic nervous system that are going to control saliva production. There's also some involvement of the cerebral cortex, so like the actual like gray matter in the cerebrum, that is relaying information to the salivary center in the medulla. And the involvement of the cerebral cortex is going to be like when you see food, when you smell food, those are coming from different sensory organs. So they kind of make a pit stop in the cerebrum, right? Because you have to see the food, you have to process that image that would take place in your cerebrum. Okay. So this is all sending information to the salivary center in the medulla. Then the medulla tells your salivary glands to make saliva. You actually make two different types of saliva. So some saliva is more watery and some saliva is more protein rich. So depending on which part of the autonomic nervous system is more active, you are going to have kind of different contents in your saliva. So watery saliva is produced by parasympathetic activation, whereas sympathetic activation, so in situations of kind of like stress, you're going to produce a more protein-rich, kind of viscous saliva. And so this is kind of why if you're really nervous, right, you might get kind of dry mouth. It's because of all the sympathetic activation. You're kind of producing less water in your mouth. You're producing this more protein-rich viscous saliva if you are producing saliva. So that's part of where that sensation kind of comes from. So from there, we're going to move to the stomach. We're not going to really expect to see anything secreted by the esophagus or the pharynx. Those are kind of just tubes getting us into our stomach. That's our next big kind of organ that is going to be releasing these enzymes and fluids necessary for digestion. Okay, so this is a picture of what's going on in the gastric glands. Not too concerned about you understanding this diagram specifically. So we're going to move on to the text part of this. Okay. So what we're interested in when we're thinking about the digestive products of the stomach, we're thinking about the secretion of acid, and we're thinking about the secretion of pepsinogen. Right, so the acid, it's creating this very acidic environment, kind of denaturing proteins, kind of breaking stuff apart, unfolding in the stomach. And then the pepsinogen is an inactive form of an enzyme. It's a zymogen, turns into pepsin, actually lets us start to break down our proteins. Okay. Fortunately, acid secretion and pepsinogen secretion, because we need the acid to activate the pepsinogen, the acid and pepsinogen are kind of going to be regulated together. So we're not going to expect to see big differences in how the acid versus the pepsinogen is being secreted, because we want them together at once, because in order for the acid to do its job, in order for the pepsinogen to do its job, we need the acid. So it wouldn't really make sense to have them doing separate stuff. So both of them are going to be stimulated. They're released by the parasympathetic nervous system. All right, rest and digest. So you should expect to see the parasympathetic nervous system highly involved in all these kind of digestive processes, right? So parasympathetic nervous system is partially controlling this. So that would be like the cephalic phase regulation of the secretion. Then we're going to have some more local hormones that are involved in releasing the acid and pepsinogen as well. And so specifically gastrin, which is one of the hormones that's being released by the stomach cells themselves, by the G cells in the stomach. And then histamine is also involved here. So histamine also helps us release acid and pepsinogen. So the cephalic phase regulation of the secretion of acid and pepsinogen would look something like: you see food, you taste food, you feel that food as you're chewing it, you feel the swallowing, you're smelling the food. These stimuli are going up to your brain. Your central nervous system is noticing this, and it's going to send out a signal through the parasympathetic nervous system to your stomach to release acid and pepsinogen, and also to release the more local hormones, the gastrin, specifically the gastrin, that are going to kind of activate the secretion of acid and pepsinogen. Okay. So if we looked at the flow through these kind of different triggers, it looks something like this. So here we see at the top, right, our initial stimulus for the cephalic phase control is going to be the sight, smell, taste of food, upregulating parasympathetic activity, so increasing parasympathetic activity. So we have a primary effect of that on the parietal and chief cells, which are to increase acid and pepsinogen secretion. We also have this kind of side pathway that tells us to release gastrin, which ultimately helps us release more acid and pepsinogen. So we also have gastric phase regulation. So the gastric phase regulation is going to be a response to the actual kind of feel of like proteins, amino acids, food in your stomach, as well as the distension of the stomach. So as your stomach expands, you have sensors that would detect that as well. So you'd have some mechanoreceptors there, as well as chemoreceptors sensing the presence of proteins. So we'd also have gastric phase regulation here, triggering gastrin, which is our hormone that tells us to release acid and pepsinogen. So we'd have a flow looking kind of like this, right? So we can see that when we see proteins in the stomach, when our stomach expands, we have sensors for that. We'll have a direct effect, releasing acid and pepsinogen. We have a kind of indirect effect on the G cells where they stimulate the release of gastrin, which is a hormone. The gastrin also has an effect on acid and pepsinogen. So importantly, we don't always want to be secreting acid and pepsinogen. Like, it would not be helpful if when your stomach is empty, right? It's been 8 hours since you last ate or something, right? You skipped some meals, you're super hungry. You don't want to have a bunch of acid and pepsinogen in your stomach trying to actively digest the walls of your stomach. So we also want to inhibit secretion. So some of that is going to be under gastric regulation. So some of it's going to be gastric phase. Some of that inhibition of secretion, so stopping the release of acid and pepsinogen, is going to be intestinal phase. So a signal coming from the intestines, which are the next stop for food after the stomach. So in terms of gastric phase regulation, it's going to be that your stomach actually senses that food is leaving, right? So the exit of food itself, the emptying of the stomach, removes the stimuli that were telling us to release acid and pepsinogen, right? So say you have your proteins, right? You ate a burger or something. They were in your stomach. They told your stomach to release acid, pepsinogen, told it to release gastrin. But now that burger is moving into your duodenum. That protein isn't there anymore. So those chemoreceptors wouldn't be sensing protein. So we wouldn't have that impetus for the release of acid and pepsinogen. So that kind of turns off that first signal. And then the actual movement of this food into the intestines, so feeling food entering the duodenum, which can be sensed by the presence of fat and acid. So we'd have chemoreceptors there, feeling that the intestine is expanding, so increased distension. That would also inhibit the secretion of acid and pepsinogen. This note about increased acidity inhibiting gastrin, this has to do with the fact that, like, as there's less food in your stomach, the proportion of the chyme, the proportion of what remains in your stomach is going to be more and more kind of hydrogen ions, right? As that food is leaving and you don't have enough stuff kind of like mixing in and watering it down. Okay, so we are going to stop here. So we are going to kind of skip this section on pancreatic juice and bile completely for the exam. It's a big topic. If you are interested, go ahead and read through this, but I won't hold you to it. And we'll pick back up on Friday. We may do like one slide on GI motility just for some basic definitional terms that you'll see in your future careers, but then we'll move directly into a reproductive system. Okay. Have a great day, everybody.
Thank you.